Okay, well, good afternoon, everyone. Let me do a couple of other things really quick, and then we will get started with the webinar. All right, well, good afternoon, everybody. My name is Crystal Parteka, and I'm the project manager for the Northern Grapes Project. Today's webinar is From Vine to Glass, Understanding the Flavors and Aromas of Cold Hardy Grapes and Wine with Fennell, Somshai Rice, and Adrian Hegeman. Anne is a professor in the Department of Horticulture, Forestry, Landscape, and Parks at South Dakota State University. She has worked with the physiology, cold hardiness, and functional genomic analysis of cold hardy grapes since 1992. She also leads the fruit genomic characterization team for the Northern Grapes Project. Somshai is a PhD candidate in toxicology at Iowa State University, where she also earned her MS and BS degrees. She works in Dr. Jacek Kozel's lab using multidimensional gas chromatography and mass spectrometry combined with simultaneous aroma analysis to evaluate grape and wine samples. Adrian is an associate professor with a joint appointment in the Departments of Horticultural Science and Plant Biology at the University of Minnesota. He is interested in comparing the metabolism of Vitis vinifera and Vitis riparia hybrids to understand the molecular basis for the difference in grapeberry color and flavor and aroma properties, as well as physiological traits such as stress tolerance. Remember that as a participant in today's webinar, we ask that you keep microphones muted if you have them. If you are having any technical difficulties, please type a note in the chat pod on the lower right-hand side of the screen, and I'll do what I can to help. If you have questions, those go in the chat pod as well. We're going to have question and answer session after each speaker's presentation, and maybe we'll have a little bit of time at the end to do any follow-up questions. You can also type your questions in at any time you have them. I'll keep track of them during the course of the presentations. So with that, Anne, I am going to let you get started.
Good morning, all. Sound good?
Yes, you're fine, Anne.
Okay. So thank you all for coming to the webinar. I'm going to be talking about one of the projects that we have not had a webinar on before, but this is the area where we're looking at the fruit ripening process, and we're comprehensively tracking what genes are activated at different stages, what metabolites or chemicals, the flavors and sugars and other compounds that are produced in the grape, and what sensory descriptors develop. Our intent with this is in part to identify genes that can be used for selection of desirable traits. And while I am speaking first, in some respects, I go first and then I come last as we go through Somshai Rice and Adrian Hegeman's work as well. There are some 30,000 genes in the grapevine genome. So the other interesting thing about that is everybody's very well aware of genomes now, since they're sequencing genomes, your personal genome, for medical purposes. The grapevine was the fourth higher plant to be sequenced, and the first genome assembly was released in 2007. That's only 4 years after the human genome was released. So, and these are two genomes that rely on each other or are intertwined in other ways. So we have some 30,000 genes that can be looked at, but we're really not interested in all of those, and we're going to focus on some specific areas today in the grapevine genome. So most of you are familiar with looking at genealogies. And the first thing I'd like to note is that if we look at Marquette and Frontenac, they do have some of their pedigree in common. Now, this is a diagram from Chateau Strip Mine, and it shows the complexity of Marquette's pedigree. If we compare Marquette and Frontenac, the area in blue, that's with the blue square, is an area that of the pedigree that's in common between Frontenac and Marquette. So if you take that area in blue and use the parent other parent Riparia, then that's how they got Frontenac. You take that area in blue, and it's back 2 generations in Marquette. So you see that there's greater vinifera in the Marquette. So this gives us a very nice starting point genetically to start looking at some of the differences between these vines and why do they have different flavors, aromas, differences in their color. So the intent of the project was to go from the genome to your senses, realizing that when we talk about wine, we also add another genome in there, which is the yeast genome, which was the first eukaryote. So it was the first, it was sequenced before the human genome. But we're going to focus strictly on the grapevine and its products. So our intent was to look at the genes that are activated, the chemicals that are produced, sensory descriptions, and then the area that we still have a lot of work to do because we're still assembling all of these parts individually at the University of Minnesota, Iowa State, and South Dakota, are how do these particular things correlate, and then from there can we identify signature molecules or compounds? Can we identify signature gene that we should be tracking for the breeder, as well as to use as benchmarking in any of, say, our viticultural studies? Sorry, I'm having a hard time hitting that little arrow that advances the slide. So in this study, I'll set the stage for this, is we were looking at over a series of time points from veraison to, in the case of, we were fortunate to have 1 year in our study where we could get very heavily ripe fruit up in the 26 bricks for both Marquette and Frontenac. And we were collecting berries weekly, taking the standard bricks PHTA, and whole berries were frozen for Zeta Vickers lab for sensory and aroma analysis. Berries were separated into skin and pulp for gene expression and metabolite analysis. So pooled samples were split up into various aliquots, and then wine was made at three different bricks points, which again was used for sensory, as well as Somshai used that for all of her aroma analysis or volatile compound analysis. So if we look over the course of ripening, Frontenac is usually about a week later than Marquette for us. You can see in the picture sort of the time frame that we were collecting samples, and then I'm going to focus on the transcriptome or the gene expression. So what genes are expressed at a particular point in time? Are they highly or expressed, or are they downregulated or shut down in their expression? And then we will be looking, Somshai and Adrian will be talking about the others. So just to give you a quick overview, there's a continuous changes in genes that are expressed. Thousands over the course of this period can be upregulated, so their expression is higher, or downregulated, their expression is lower. And in general, there's a greater number of genes that are regulated one way or the other in the berry skin than in the berry pulp. And this says, whenever you're doing a gene expression study, is it logical? Is the results that you're getting logical? And this is kind of logical. We do know that the berry skin is a primary source of many of our flavor compounds, as well as our colors in our wines. So if we do, it's a fairly complex study, and so I'm not going to cover all of the time points for this talk. I'm just going to focus on some key points at harvest. So one time point in the study. So the number of genes, what you see in this diagram, in the figure A, we have two circles, one that's labeled Frontenac and one that's Marquette. And in the outside of that, you see about a similar number there. And that is genes that are specifically expressed in Frontenac or Marquette, but not in the other. In the center, in that purple area, you see there's about 3,000 genes that are expressed differentially. That means they're either up or down regulated, but they're expressed in common between Frontenac and Marquette. So if you look back to that, that's something that you would expect. You've got some pedigree and background, so you expect to see some things in common and some things different. Now, if we look at the B diagram, where you have skin and pulp, you can see that we have a greater number of genes that are differentially expressed, and this is across both genotypes in the skin in comparison to the pulp. And then some of these are in common. And so what we can do just by these two things is we're seeing, okay, yes, it makes sense. There's differences between the cultivars. There's differences in skin and pulp. And then we can start looking at what are those particular genes and what are they involved in. So what I'm going to focus on is two components which everybody's used to seeing and looking at in their wine or in their juice or in their berries. And one is the color development. That's one of the things that we track in the field when we're looking for harvest parameters. And the other is aroma development, particularly the terpenes or the terpenoid biosynthesis. Now, terpenes contribute to scent, flavor, color. They were originally discovered in the late 1800s in pine resin, and they're the compounds that are very important in terms of flavor and aroma in many plant products that we eat. And so these are particularly important in the wine world, and so I'm just going to use these two as a case study. So first off, does the gene expression coincide with existing knowledge? And one of the things that we note is in doing the data analysis is that the anthocyanin biosynthesis genes, these are the ones that are specifically involved in making the color compounds that we can see, are preferentially expressed in the berry skins in both Frontenac and Marquette. People often look at the colored flesh of these two cultivars and think they are Teinturier type, or which means dyed flesh, but they are not true Teinturier types of grapes. Most of the color develops in the skins and then appears to leach or move slowly into the flesh. So one of the things that we know is that Frontenac, being a black grape, it has a very dark juice. And if you just look at this little chart that I have up here, what I've listed is about 6 genes in the anthocyanin biosynthesis pathway. And what you'll note is there are some of them, the darker the red, the more highly expressed that gene is. And we're looking at some genes that are pivotal in anthocyanin biosynthesis in this little diagram. And so what you note is a great deal of higher expression in Frontenac than in Marquette, which would contribute to differences in the color development. And Adrian will be talking a little more about those in a little bit. So the next question is, are there any differences that are related to aroma or flavor? And so we immediately look to the terpenoid biosynthesis gene expression. The marvelous thing about grapes is that we like them, and so we have been studying them for a long time, and we do have a fair number of compounds and genes information in this particular pathway. There's over 500 genes in the terpene biosynthesis pathway. And I don't know, Adrian can probably give you a better idea of how many compounds we've identified specifically in grape. But what we would like to know is the specific genes that are involved in the development of specific flavors or aromas. So again, verifying some things that we know that there's a greater number of gene expression differences in the skin than in the pulp. We know that the berry skin is the source of many of our aromas and flavors, so this is logical. The expression pattern, though, between the two cultivars, so if you look at the first column in this chart, those are all specific gene identifiers. So these are some of the 30,000 genes in the grapevine genome. The next column is the expression level, how highly expressed or lowly expressed the particular gene is in frontenac skin, and the far right column is the marquette skin. So I've just colored it. Dark red is the more highly expressed. Pale pink is the most lowly expressed. And so you just looking at that, you can see there's differences between marquette and frontenac. The interesting thing becomes when you start looking at specific genes in relation to flavor compounds. And so in there, for instance, in this case, I've highlighted one gene that's highly expressed in marquette, and this beta-ocimene synthase, which contributes to that woody green and vegetable nuances that are sometimes detected in marquette. In contrast, one that's highly expressed in frontenac is the pinene synthase, which gives you some of that eucalyptus camphorous notes with the spicy pepper and nutmeg nuances. And where we're going with this in the future is with Somshai has been identifying through chromatography and sensory analysis specific compounds, and then we can combine these two pieces of information together to highlight the specific genes that are involved in those particular the synthesis of those particular compounds. So, as a quick overview here, there's distinct cultivar differences in the gene expression patterns. There's distinct differences in genes related to aroma and flavor that are found between marquette and frontenac. And what we are going to do next is start correlating these with the volatile compounds, as well as the other metabolites that Somshai and Adrian have been analyzing. And from there, I'm going to sign off. This is a number of the people that have been involved in the Northern Grapes project in this particular fruit ripening and composition study.
Okay, we have one question that I don't know if it's something that you'll be able to answer, Anne. Tom asked, how does oak aging methods help or hurt these grape wines?
Okay, so there you start getting into an area that I think is more in the line of Anna Catherine Mansfield's area. In terms of that, the I'm a genome geek sort of here, so I go from the genome to the field and back, and I'm very interested in determining those compounds. So you're taking things a step down the road, and I'll have to defer that question to see if Adrian can chime in on that one.
Okay, how about we just leave that one tabled since it is more on the winemaking side of things. And Patrick had a question that you would probably be more able to answer. And he asked, interesting that you mentioned the pinene eucalyptus-like aroma component in Frontenac. I've picked that up a number of times in our Frontenac wines. I'm not sure it's a desirable aroma. Does it diminish with further ripeness, or can it be masked somehow?
Where I was seeing that was in our rather jammy Frontenacs. So that, until we get all of the time points back and compare them with Somshai's analysis, I can't tell you if it's an early in ripening or late, but definitely in what I presented just there, those were the rather jammy Frontenacs that we were detecting that in, and it was a comparison between Frontenac and Marquette. And so it will be really interesting to see in terms of the time points, because Somshai got those time points, to see if she noted it increase or decrease at all.
Okay. Then Tim asked, he says, anthocyanins, vinifera color stability is related to the particular anthocyanins, monoglucosides, while many hybrids have various diglucosides. This affects the hue of the wines. What specific compounds did you find? Anna Catherine's student mentioned Frontenac, St. Croix, and other northern grapes cultivars are dominated by delphinidin 3,5-diglucoside and malvinidin 3,5-diglucoside.
And I think Adrian will address some of this, but yes, the diglucosides are more dominant in the hybrids, and it's actually one of the ways that since you're not supposed to be using hybrid grapes in Europe, it was one of the ways that they used to determine whether or not hybrids were being used in wine. And it definitely has an impact on the color. In a separate study with the VitusGen, which is another USDA project, we have been able to identify specifically the inheritance of the monoglucosides from the diglucosides. And so that's one thing, because the diglucosides tend to brown more than the monoglucosides. That is one thing that we've got some pretty good gene identity for the breeders to start using in their selection process.
Okay. One last question from Corrine before we move on. She says, in my group, we carried out volatile compound analyses in Frontenac and Marquette and found much more terpenes in Marquette compared to Frontenac. But according to your transcriptomic analysis, it looks to the contrary. Can you comment on this, please?
The specific thing there, you're talking at gene expression versus the actual identification of the compound. And you have to remember that Marquette and Frontenac don't ripen equally. And so we're not quite comparing in this study, which was rather time dependent, where Marquette is already at a very overripe phase. So it may have been diminishing in those compounds in comparison to the Frontenac. So that's one key point there. You have gene expression, but then you have to determine how that relates to the actual production of the product. So without the whole time course, I can't fully answer your question because you were seeing one time point only for each cultivar.
All right. Thank you, Anne, for the presentation. We did have one other question come in, but I think we're going to just go ahead and we need to move on to Somshai's presentation. So Somshai, I have your slides pulled up and have given you control of them, so I'll let you get started. I think you're still muted. So
Okay.
There you go.
There we
Got it.
Okay, I am going to speak about the chemical and sensory analysis of the aroma profile of Frontenac and Marquette. And very quickly, I wanted to take you through the tools that we used, the solid phase microextraction tool, multidimensional gas chromatography, mass spectrometry with simultaneous olfactometry. And we used these tools to measure VOC, which is volatile organic compounds, as in vivo volatile metabolites of these grapes during development and all the way through to maturity. We also used these tools to evaluate the wine made from Frontenac and Marquette, harvested at 22 and 24 Brix, using an aroma dilution analysis method. So I will kind of breeze through the berry results. Those are available in the progress reports linked in the Northern Grapes webpage. So to begin, this is a solid phase microextraction fiber. There is a 1 centimeter fused polymer coating on the bottom there that's sitting inside the cylinder. And that is just very simply an air sampling device that works like a syringe. You expose it to extract the volatiles, you retract it for transport and analysis in the lab. And that advantages of SPEME are that it's very simple, it's reusable, you don't have to use any solvents to pre-concentrate or extract VOCs from your sample. We did take it into our lab here at Iowa State University. I would just call it MDGCMSO from here on out. The acronym is on the bottom in the caption there. So this is Dr. Yashikozio's lab. He is front and center in the picture there where we carried out the simultaneous chemical and sensory analysis. So I like to say that our instrument is pretty standard. It's a standard issued adjuvant. But inside, under the hood, it's more like a Formula One car. If I can call your attention to number 15, the coil on the left, that is a nonpolar column that separates the volatiles collected on the SPEME and exposed in the hot GC inlet. It separates the volatiles by boiling point. Immediately connected in series, number 16, is the polar column, and that separates the VOCs by the chemistry of the VOCs. And so this is where we get the 2 dimensions, the multidimensional gas chromatography. Another added bonus of this system is that it allows for simultaneous chemical ID by mass spec. So if you look at number 17, that horizontal line going to the left into the MSC, that's the mass spectrometer. At the same time we are IDing the chemicals, we can ID by olfactometry through the sniff port, which is number 22. And by olfactometry, we generate it's a user-generated aromagram. We characterize these volatiles by aroma descriptor. We can characterize by hedonic tone and also the intensity of each aroma note of the sample. There are 2 ways we collected volatiles, a non-destructive and a crushed berry destructive analysis. So the picture on the left, we are using a polyvinyl fluoride film, also known as Tedlar, trademarked by DuPont, to enclose the grape clusters. And the picture on the right, we inserted a SPEME fiber exposed to the polymer coating to pre-concentrate the VOCs for a certain amount of time, retracted the fiber, and then analyzed them in our lab. We also tried a second way that you are looking at a modified 2 milliliter glass vial that we chopped the end off, not we, our glass blower, chopped the end off and flared the edges a little bit, and we pulled a vacuum just with a syringe and needle to hold it onto the grape, and inserted the steamy fiber through that same hole to extract volatiles that way, to further isolate the berries from, say, any volatiles that might be around in the air or emitting from the grape leaves themselves. These fibers were then taken back to our lab for analysis on our instrument. And this is Jason Vallone. He is an ISU graduate in chemistry. At the same time, we did the in vivo volatile metabolite analysis, the non-destructive. We did randomly pick berries from these grape clusters to crush and analyze volatiles in the laboratory as a kind of like something to benchmark it against. So we are looking at data here from SDSU, Marquette, in the in vivo analysis from vibration to harvest. This is the non-destructive in vivo sampling. And I just want to draw your attention to, I know it's a lot of data, just draw your attention to the vertical axis, how high up something is on the vertical axis can be related to concentration, and also the number of compounds detected, which is 7 on the left-hand side there. What we can tell you is that we detected a lot more in the crushed berries than the non-destructive in vivo sampling, both in concentration and in number of compounds detected. So you were looking at, let me go back here. So this is Marquette in vivo, Marquette crushed. This would be Frontenac in vivo, Frontenac crushed. So again, I'm not going to comment much on this except for we do have plans to link the gene expression with Anne's data regarding the metabolites detected. Next, I did a aroma dilution analysis using an automated steamy method that we developed in our lab. And in a nutshell, we analyzed the wine samples neat or undiluted, and then I diluted each sample successively, 1 to 1, 1 to 4, 1 to 8, and so forth, with a model wine that was pH buffered. And the idea here is that with each dilution, the compounds with the lowest aroma impact dropped out of the picture, or they were not detected at the sniff port by a human panelist. The last compounds standing we deemed were the most contributed most to the overall aroma of these wines. So what you are looking at here, the red trace, the red signal is the total ion chromatogram. So that's the chemical analysis coming from the mass spectrometer. The black trace is the aromagram, which is the user-generated signal, where we describe the aroma events by descriptor, intensity, and hedonic tone, as I stated before. So you are looking at Marquette wine made from 22 degrees bricks, neat, no dilution. And I'm just going to focus on this as a case study because there's a lot of time points to go through. But the take-home picture, or the take-home message of this figure is there were 15 aroma events. So if you look at the black trace, 1 through 15, there were 15 aroma events in undiluted Marquette wine harvested at 22 degree bricks. At a 1-to-1 dilution, which is what you're looking at here, we dropped down to 11 aroma events. At a 1-to-4 dilution, again, of 22 degree bricks Marquette, we dropped down to 4 aroma events that still hung around. And then at a 1-to-16 dilution, we have 3 aroma events. So we went from 15 to 11 to 4 to 3. odorous compounds. So what we found in this very brief experiment was that the Marquette wine aromas developed from kind of like a wine, apple, and fruity aroma to a cheesy chocolate strawberry type aroma by our human panelists. The chocolate and molasses notes and aromas intensified, and a jammy aroma developed in Frontenac wine as the sugar levels increased for this batch of wine. So I found out a lot of chemicals, 34 to be exact. These are the red. Yep, they're red. The red dots in this principal component analysis plot. So all you're supposed to take home from this picture is that there are a lot of rose, apple, banana, fruit type flavors. So aromas in these wines. So if we were to overlay where these wines fall chemically, it would look a little bit like this. The Frontenac, the Marquette wine harvested at 24 bricks had higher relative concentrations of a chemical that would give it that waxy pineapple, banana, apple peel kind of aroma profile, and lower in like the raspberry pear profile. Okay. The Frontenac it's the lower right-hand quadrant there. The Frontenac harvested at 24 degrees bricks has more of a tart, fruity butterscotch with some peach and pineapple in there, and very, very low in minty or peppermint type of aroma. And just visually looking at it, you can kind of see that the wines made at 22 bricks, both Frontenac and Marquette, would be high in the fruity pear, banana aromas, almond, burnt sugar, chocolate, and peach is what I got, and then very low in all of the other chemicals found in the upper right quadrant. So I'm going back here in this upper white quadrant there, are not highly associated with wines made at 22-degree bricks. So visually, you can kind of see that where they grouped according to volatiles. But we've verified this with a hierarchical cholesterol analysis, and you can very clearly see that they divide into 3 distinct groups, with the 22-degree bricks wines being the most similar, even though they were made from Frontenac or Marquette, and the 24-degree brick wines were very different from each other, and also from the lower sugar wines. And that's pretty much what this last slide is summarizing here. We are continuing this work. Similar work is being done. We did a lucrescent skin contact study to see if flavor and aroma profiles were affected by skin contact. Also in Edelweiss. And then I just got some Brianna and Frontenac Gris from Anne. So we're going to continue this work in a similar fashion. And I think that's all I have. Any questions?
Okay. Yeah, thanks, Somshai. We did have a couple of questions come in. The first one was from Tim, who says, many of the terpenes contributing to wine aromas are bound terpenes that don't contribute to aroma until they are released during winemaking. Mm-hmm. If they are bound in the whole or crushed grapes, what do headspace aromas of grapes in the field tell us about what we could find in the wine?
The volatiles from the crushed berry analysis, I didn't use any enzymatic activity to release. If they were glycosidically bound to glycosides, this is more, I would say, a free volatile analysis. because there wasn't any, I didn't control for any enzymatic activity.
Okay. And then Tom, when you were looking at the figures like around slide 19 and 20, asked, where's the spice and tobacco? I think you wanted to know like where on the figure those appear.
I have that, but not in this slide. I think is that something we can table and I can provide to you?
Yes.
Later. Okay.
Yes, Tom. So send me an email and I can get that information to you, please. Let me see here. Then Corrine asked if she says, "May I ask what was the fermentation temperature? It may have impacted the level of isoamyl acetate, banana aroma, and ethyl acetate, pineapple you may have found in your wines."
I will actually. This question would be directed at Anne. The wines came from her, and I don't know how they were made.
Anne, if you're on and want to answer
temperature, these were small batches of wine, so there was no temperature control in the fermentation process. That the winery does use controlled temperature in its standard fermentation, but these were small batches. So it was the winery temperature that they were produced under. And I will tell you that that winery, their facility goes back into the Black Hills, and so it's a fairly stable temperature in their winery. It doesn't fluctuate very much at all.
Okay, thanks, Anne. Let's see here. We had another question from Tim, which Anne's probably going to have to answer this. Did the wines have any standardized acidity and residual sugar? How are they standardized? You may have asked that. To answer that, I was looking through the list to get all the questions and not really listening.
Okay, this was the first study we did, and so in the discussion, the choice was made to take them at specific Brix points or time points. So basically, they ended up being a week apart. In Frontenac and Marquette, in the Marquette, in the Brianna and the Frontenac Gris, everything was adjusted. So when those were taken, and we went to a standard sugar content, so that we would have a fairly standard alcohol content. In terms of what the residual sugars were, I don't have that information in front of me, but we did send everything to Merle, and they did an analysis of the wines that were sent to both to Minnesota and Iowa State for the VOC analysis and the sensory analysis that were done by Emily Del Bell. So I guess I'm saying wait for the publication.
All right, thanks. Somshai, and thanks, Anne, for jumping in to answer the questions. I think we have addressed everything, and in the interest of time, we had probably better move on to Adrian. So, Adrian, I see you have unmuted your microphone, and it looks like you are ready to go.
Yes, I have. Can you hear me?
We can, just fine.
Excellent, excellent. Well, good morning to some of you, and good afternoon to the rest of you. So I'm going to try to finish this up. I may have to skip over some things in the interest of time. I'm really, let's see, slide advancement. There we go. I'm at the University of Minnesota, and I work with Jim Luby fairly closely, but most of the work I'm going to show you today is done by a graduate student who's been very productive, Soon Lee Te, and a undergraduate technician, Betty Rostandy. So our goal is really to do what we can to try to make connections between the type of information that we've heard of from both Somshai and Anne, so that we can then, really understand what are going to be useful tools to guide breeders via marker-assisted selection to, identify those genes that are going to be associated with desirable or undesirable metabolites that affect pigmentation, organoleptic traits, aroma, et cetera. And one of the challenges, of course, is that these are complex traits in many cases, and there's not a one-to-one interaction between a perceived aroma or some one of these characteristics, but it's in fact a combination often of things, or there can be, for example, co-pigments that affect the anthocyanins we've been talking about in ways where we really need to be able to perform a statistical analysis that looks at large populations of molecules and understands how things may be influencing each other. And, for example, in the case of aromas that we were just going over, many times aromas, if you have a pure compound, you can get a nice one-to-one association between that chemical and a specific smell. But if you start to mix together different aroma compounds, you start to create more complex characteristics. So that are harder to work with because of that complexity. So I'm going to show you a little bit of what we've done with Frontenac and Marquette, looking now not at the volatile compounds that Somshai has done a good job of talking about, but now looking at some of the chemicals that are not volatile, that are still going to be important for modifying aromas and participating in these organoleptic and pigmentation characteristics. So let's see. Having issues with, there we go. Okay, so what sort of compounds am I talking about? I've included the chemical structures here for those of you that are aware of them, but just if you're not comfortable with chemicals, you can look at these and see similarities in the shapes and so forth that are important and informative. So the first class I have outlined here are the organic acids. It shows the structure of malic acid, which is a big issue in many of our grape varieties, and then tartaric acid. You can see these are very similar acids, and structurally they're quite similar. I'm actually not going to spend a huge amount of time talking about the organic acids today, but more want to focus on these other two classes. In yellow, these polyphenolics, which include chemicals such as resveratrol, which has been very popular in press and medical releases as an important chemical in red wine for human health. It's been associated with the explaining the French paradox, for example. These other compounds, quercetin, polydatin, or other polyphenolics found in wines that are important for their antioxidant properties. A similar class of compounds to the flavonoids and polyphenolics are these anthocyanins that we've been talking about that are responsible for the pigmentation. And this shows the sort of core structure of the anthocyanin is made of these hexagons, a sort of chicken wire structure. And then when we were talking about the different sugars attached, this is a mono gluco anthocyanin, malvidin. But you can also attach more sugars around this core structure and get these diglycosidic forms that Tim was talking about, or have ones that don't have sugars attached that also contribute to the color. And there's a little chemical analysis shown here, and I can explain how this works. Basically, we can separate these anthocyanins into individual chemicals. Each chemical will have a peak associated with it, and you can see I have an analysis of Marchal Foch, Frontenac, Frontenac Gris, and Marquette shown here, normalized to the weight of the berry material. So you can see, obviously, the Gris has much less of these pigmentation compounds. You can see the Frontenac has significantly higher intensity of these anthocyanins than the Marquette. But the actual anthocyanins, in many cases, are fairly consistent across these. And this may be consistent with the fact that they have and share much of their pedigree, but some aspect of the regulation of the total amount of pigmentation is causing there to be a higher level of pigmentation in Frontenac versus Marquette in this case. Okay, so this is a slide that Anne showed you. I wanted to sort of put us into context. She talked about the genes that were being produced in the transcriptome and talked about and showed data from these the gene expression information. We're looking then at the next step, this metabolome, or what are the chemicals that are being made by the genes within the grape berry during ripeness. And then Somshai obviously talked about the sensory descriptors. So we really need to have measurements of these chemicals that we can relate statistically between the gene expression and the sensory descriptors, so that we'll know which genes are going to be important for guiding our breeding selection. If you have to wait for berry production before you can evaluate your individuals within the breeding programs, that's a huge investment in time and field space. So if we can learn something about the genes that are going to be important for specific sensory descriptors through this analysis, we can take those back to the breeders and give them a set of tools where they can actually screen grapes at a very early stage before they're capable of setting fruit, and use those to really understand what their potential is for having the different sensory descriptors and traits that we're either interested in or want to get rid of in some cases. So, our workflow is, I'll just dwell on this briefly. We take the grape berry material, as Anne talked about, and split it into skin and pulp and seed materials. These we extract then with ethanol, which does a very good job of getting out many of the compounds. During a homogenization process, we can then go through various other steps. And then we use, instead of a gas chromatography setup, we use a liquid chromatography, mass spectrometry analytical setup that allows us to look at things that are in solution. So these are going to include big compounds that may not be volatile, like the ones that Somshai talked about. So this is just an example of the data. Again, you get peaks that are associated with each of the chemicals, and then we can make comparisons between time points in order to understand how chemicals are changing over the analysis time. Here's what we call a mirror plot, where we take the peaks from a Marquette analysis shown on the left, and so those peaks are increasing from the center off to the left, versus Frontenac, where they're starting at the center and coming off to the right. And what you can see is that there's actually quite a bit of similarity in the major compounds that we're seeing through this analysis between Marquette and Frontenac. Many of the diglycoside anthocyanins are actually coming out in these early peaks, but we can see some big differences, for example, in this caffeic acid peak at the beginning, where we have much more of that in the Marquette. This is an aromatic compound that may, in fact, be interacting with the anthocyanins and functioning to some extent to modulate their properties. If we look at the monoglucosidic anthocyanins in these two samples, we actually see that they're fairly similar abundance on this plot. So you see the delphinidin, the peonidin 3-glycoside are fairly similar between Frontenac and Marquette. These are similar simply, as I pointed out earlier, because the same similar chemicals are being made in these two species. Frontenac really just has much more abundance, and for this figure, they've been normalized for the purpose of comparison. Where we see big differences are often in things that aren't pigments, like this caffeic acid, and also these other flavonoid glycosides like myricetin. But there are some other subtle differences in the peaks that are shown up here that are kind of hard to get into. But we'll be able to take this data and basically convert it into a set of numerical scores for each of these developmental cases, and then compare this using statistical techniques with Anne's gene expression in order to try to find out what are correlated in terms of the changes that we see here in the anthocyanin levels and the genes that seem to be different in her analysis. And that'll allow us to get some information about these differences. So, she showed you this as well, that obviously there are differences that we see in the genes between the Marquette and Frontenac skin expression in anthocyanin genes. And so we're hoping to be able to take this information, then go back and look at the different sets of anthocyanin glycosides, mono and diglycosides, as well as the monomeric anthocyanidins, and get information about which specific genes are going to be important for giving us the specific differences in coloration. Okay. So we're getting short on time. I wanted to just briefly touch on also these stilbenoids, and this is something that starts to get out of just into the area also of disease resistance traits. So I don't want to dwell on it a huge amount, but I did include some additional slides in my presentation to sort of show some of the work we've been doing to characterize some novel compounds in this class that look like they're likely to be important players in powdery and downy mildew resistance, potentially, as well as other fungal resistance. And so we've been putting a significant amount of effort into trying to understand the genes that go into production of this class of compounds as well. But I think in the interest of time, I'm not going to go over those slides, but you can look at them, and you have my email address, and you can contact me if you have other questions regarding that stuff. So, Chrislyn?
It's okay if you want to keep going, Adrian.
Yeah, we're okay going
need,
over the hour?
Yeah,
okay.
Go ahead. As long as
okay.
you don't have to be anywhere, I don't have to be anywhere. So,
okay. Well, I will then forge ahead at least quickly. I don't want to take up everyone's time here. But so just to go into this class, I'd mentioned before that this includes resveratrol, which is very interesting because of its effects on certain metabolic regulatory enzymes in human and animal health. It's been associated with increases or beneficial effects in cardiovascular health in humans, as well as effects on mitigating the aging in some cases. So it turns out this is a really interesting family of compounds because particularly grapevine makes a lot of modifications of these chemicals, and they're often in response to infections and don't appear until a disease state is showing up. So it makes them a little bit more complex to study, but many of them are present even in undiseased grape tissues. So what we see, though, is that they seem to be, in a couple of cases, there's some information that some of the more complicated stilbenoids may play a role in killing off certain infectious agents, particularly the downy and powdery mildew pathogens. In particular, there's been some work highlighted in these red boxes here showing that this hopiatinol and viniferin are fairly effective toxins against the causative, one of the causative fungal pathogens. And so these have worse, more complex structures. These are real chicken wire structures where you've now taken that monomeric resveratrol structure shown here in the upper left-hand corner and basically stuck a bunch of them together in order to make these new compounds that have activity against these pathogens. And furthermore, when we start to look at the appearance of this class of compounds in the cold hardy grapes, we see quite a bit of variation in which of these are expressed between different individuals. So we're really interested in seeing what extent we can tie these into disease resistance characteristics within these populations. Just I included the process. We actually, obtained, I should say, Soon Lee obtained a huge amount of the cane tissue from pruning, which is a good source for these compounds from John. And then we were able to go through this procedure to try to extract these compounds and figure out their structures. We're interested actually in the fact that these may be a good source for other uses as well, and so we are exploring whether or not we could in fact create a economic value for grapevine cuttings, because if we can extract chemicals that can be used as antimicrobials, that could be useful as, for example, an organic fungicide, or it could be used for a preservative that's organic for the cosmetic or food industry. So we're currently exploring those options as well. But this process involves extraction and grinding up the cane tissue, and then basically separating the chemicals on long columns into separate different chemicals, and then analyzing those by mass spectrometry and other techniques to figure out what their structures are. So that was the additional material I wanted to go over on top of the fruit information. So I guess I can take any questions now.
All right. Well, we have one come in so far. First off, thanks,
Sure.
Adrian, and sorry to those of you that we ran over a little bit, but just 1 minute over, so that's not bad. Tim says that hypothesis has been put forward by Gavin, that's Gavin Sachs' group, that some of these compounds associated with disease resistance are also associated with low tannin extractability. So I guess that's more of a comment than a question.
Yeah,
that's probably important to talk about.
That's a really, really good point. And one of the interesting things about these is there's classes of compounds is that they interact with each other, and they also form very strong interactions with proteins. So it's one thing to extract them out of the plant material and then characterize them isolated, but in the actual berry and cane and leaf tissue, there may be associations between the interactions of these that are going to be really important for their actual activity. So.
Okay. Tom asked if the cane extractions have been used in biodynamic preparations, so biodynamic farming.
They have not. We're still really at a stage where we've been exploring what types of organisms they might be effective against. So we have run some antimicrobial screens in the laboratory against gram negatives, gram positives, filamentous fungi, yeast strains, as well as looked at some of their activity against the powdery and downy mildew varieties. So, we're still at the very earliest stages. Many of these compounds, because we're looking at really novel genetic material that hasn't been well characterized for stilbenoids, I think there's a lot of work to just figure out what the new chemicals are that are there. And figure out which ones are active, and then we can proceed more intelligently with what are going to be the best procedures for extractability and generating something that might be a productable extract for that sort of application.
Okay, awesome. And then Tim had one more question. How does what you found in the canes compare with what you found in the fruit?
We see way more stilbenoid diversity in the cane tissue right now. We don't see the same levels of these higher order viniferins, for example, in the fruit tissue yet, and we've looked. We do see resveratrol, we see piceid, so we see some of the monomeric stilbenoids and their glycosides, but we don't really see these higher order ones, which are often associated with the antimicrobial activities.
Okay, and then let's see here, Tim. Let me go back to a question that was asked right after Anne's presentation. It was, will gene expression change with soils, temperature, and chemicals? I think this is kind of an important thing to ask.
Yes. So I mean, this is one of the things, and not only will gene expression change, but there are concerns. There are levels of regulation that occur after gene expression that will change. So the value of this study is that we've, Anne has taken, the berry material that she's generated and taken very good care of that material and split it up and sent it off to these different analytical approaches. So we get the sensory, we get the metabolite analysis and the gene information on the exact same samples. That allows us then to apply these statistical analyses in order to be able to understand how these things are related. If we had tried to just duplicate the same experiment in 3 different locations, there'd be so much noise, you'd never be able to get any useful information out of it. But because of this split of the same samples, we can actually do reasonable statistical analysis.
Yeah, and then there's one last question from Tim that is for everybody, all of our speakers, and he says, what are the challenges in putting all this information together from expression to sensory? How do you filter all of this data? So Adrian, since your microphone's on, if you want to go ahead and take a stab at it, and then Somshai and Anne, if you want to chime in, feel free.
There are a bunch of challenges. One is, I mean, we have good multivariate statistical approaches, for example, in place for our data sets. Somshai has good data processing tools for her data sets, and Anne likewise. But the field of sort of systems biology is still working out how to deal with integrating data sets of different qualities from different laboratories. And I think the sort of theoretical statistical framework is in place, but there are a lot of challenges associated with moving large data sets around and working with them that we're looking forward to having to explore here shortly. Also, there are other challenges that we didn't quite get into, even though this was one of the earliest plant genomes to be sequenced. There hasn't been a huge amount of effort put into really understanding the function of a lot of the genes. The function is often derived from, by extension, from related species, and oftentimes that process is quite error-prone. So we've noticed, for example, there'll be, you can tell that a gene is a terpene, is involved in terpenoid synthesis because it's similar to lots of other terpene syntheses. But the subtleties of which terpene you're looking at is harder to get at. And so this is where this sort of statistical analysis, where if we see a big change in a specific terpenoid chemical and a big change in a gene from that same sample, we now have the power to get better annotation about what that actual gene is doing. Whereas just looking at the genome, you would be stuck sort of with this annotation that was based on this extrapolation from other data, which isn't as accurate. So.
Awesome. Somshai or Anne, you want to comment?
I would just like to say Adrian's response pretty much covers my thoughts on the difficulties. We are we're pioneers in this area. We don't know what we don't know, I suppose. And it's more we're just casting the net out to see what's out there. And so, I mean, that's a good pinpoint of the big problem is how to put this all together.
All right, let's see if Anne has anything to add. If you do, Anne, your microphone is muted.
Sorry. It's unmuted now.
It is.
As Adrian said, we're using guilt by association to determine some of these. If you go back to one of the slides I had early on, I think in like my second slide, there was a little diagram in there that was taken from actual berry data. and you'll see a whole pile of genes there and one compound. And so if you look at that, you were on the right one.
Oh, that one?
Nope,
that
this one.
Okay.
So you see it says metabolite genes.
Yep.
So the little red circle is the metabolite. And then we've identified a whole series of genes. Orange is something that's on in the seeds, red is in the skin, and I think pink is in the pulp. But like I said, there's over 29,000 genes in there. Exactly which ones are involved, and it can be more than one involved in producing that one metabolite. And then, as Adrian said, there's a lot of post-processing things after gene expression that can take place. So we really are, as Somshai said, this is an area that we're trying lots of different things and to define the function, because typically we have the function of about 50% of the genes well defined. We have them well characterized. That leaves a whole lot of information out there that we're still trying to suss out.
All right. Well, thank you very much for your presentations, and Somshai and Adrian. They were very interesting. I think we had a good audience on today as well. I did post a link to the post-webinar survey. We do value your responses, and we actually do use that data. So if you would take 5 minutes and go take that survey, I would really appreciate it. Let's see here. Tim, would you like to say anything? Your microphone, there you go. I'm not hearing you, Tim. So anyway, I guess I do want to make a note that this was our last Northern Grapes Project webinar, at least for our first round of funding. We are looking into applying for more funding, so just, I guess, stay tuned. So thank you very much, everyone.
Thank you, Kristen.
Thanks, Adrian.
Thanks, Kristen. So we're on again at 6?
Yes, 6 Central, 7
Okay.
Eastern. So yes, yeah, and
Okay.
you have the link. So yep.
Okay.
Thanks, everyone.
Thanks.
Can you hear me now?
Yeah, I can hear you.
Okay.
Yeah, Tim.
Yeah. I almost clicked exit.
So Tim, just real quick on some standard varieties. There's some work like this that's been done in Europe. I know of one study in Italy where they also did a lot of looking into things like slope and training effects as well.
Yeah.
So I can't remember. I think Fennell gris was one of the ones used.
Yeah. Well, I think this is a great start, and it's like casting this big net, and 3 different nets, and finding out where all the nets are correlated. So so this is really cool, forward-looking work.
I think the data has several vintages in it.
Yeah.
The other thing was in your earlier question about standardizing things, that we sort of went in the Marquette and Frontenac, we went more from what the sensory people were asking.
Yeah.
And then in the Frontenac Gris and the Brianna, we discussed it again, and since since these were aromatic, we were looking at an aromatic type grape. We decided that we would just hold as much as possible the sugar and consequently the alcohol content standard.
Yeah.
And then we're looking at the changes in the compounds with those 2 things standard in the vinification or fermentation, as opposed to the other. It was definitely one of those where we learned a lot, and I think if we're going to pinpoint an area again, we would need to discuss all of those things with what we've learned, Somshai and Adrian have a lot that they can contribute in that area so that we can do phase 2 that would provide some of the information that you're asking for.
No, that sounds great. And this is something I'm going to send this recording to Gavin and ask him what he thinks about all this stuff. So I'm sure he'd have a lot of really great suggestions.
He would.
Yeah. Okay, thanks. I'm gonna sign off here, but I want to thank all of you for doing this and being part of the project, and hopefully we can write all this up and you know, decide on where to go next on the next iteration of the grant. All right?
Yep. And we have a meeting in a few minutes.
45.
Okay. I'm gonna grab something to eat. Chris,
All right. See ya.
Bye-bye.
Bye.